major toxigenic Cyanoprokaryota orders. Now, I mentioned that there's significant taxonomic changes that have taken place with cyanobacteria. This is where you want to pull out the lab manuals. I don't have one up here, but if I remember right. This one right here. Let's look at the table of contents. Pages 1 through 16, just some general background information on algae and cyanobacteria, mostly pictures and some terminologies, so we can move beyond that. Pages 17 to 24 is the most recent taxonomy of the cyanobacteria. And of course, when it comes down to it, we're going to jump straight to the key. But the most recent taxonomy of the cyanobacteria is based on those three big books that I mentioned, Komarek. He's a Czech, very nice guy. Obviously looks in the microscope 24 hours a day, 7 days a week. But he spent his entire life revamping the taxonomy of cyanobacteria. And so his 3 volumes are listed there. And then he condensed those 3 volumes into a couple of very useful papers. And one of them, which starts on the next page, 18, is called Taxonomic Classification of Cyanoprokaryotes Using a Polyphasic Approach. This, which is 35-40 pages, and I didn't print out all of the pages. I just printed out the introduction so you get a flavor of how these things are done. And then I did put in there the appendix. So if you go to page 21, there is the appendix that summarizes all of the genera of cyanobacteria. All right? And if you look at, for example, page 21, you'll see the order Cyanobacteriales, and then Chroococcales is in the darker hatching, sometimes not that easy to see, and then underneath it are the main families, and then underneath that, the main genera. And what I have done, wherever you see an asterisk that I have put in there, those are the genera that we found to produce toxins, cyanotoxins. So if we look down within the Chroococcales, the only thing we see is Cyncococcus. And that is courtesy of the Salton Sea. When we did our study on the Salton Sea. This is a picoplankton or a nanoplankton. They're about the size of bacteria, but they are prolific producers of microsystems. And may or may not have been responsible for some of the mass bird die-offs that we had back in the 90s when we were starting to decide what we're going to do with the Salton Sea. We're going to drain it or we're going to clean it up. And then we also see Cynicocystis in that same order grouping, and a few of them that are known toxic. But the main ones that we want to focus on, go to the next page, page 22. You'll see under the Chroococcales order, the Microcystaceae family, and of course, Microcystis. That's a key one. And then over to the next page, under the order Oscillatoriales, Microcoliaceae, Planktothrix, which is the filamentous one that's producing all the different toxins, Trichodesmium, which is a marine genus that produces toxins. And then under different families within that, Oscillatoriaceae, there's Limbia, Oscillatoria. The main one, if you flip to page 23, top column on the right side, you'll see Nostocales. That's another order that we have. And within that order, if you go to the next page, the last page, you'll see under Aphanizomenon all of the main ones. These are the main ones that produce toxins. the genus Aphanizomenon, genus Cylindrospermopsis, Dolichospermum, which is the new name for Anabaena, which I'll get to in just a minute, Nodularia, Chroococcales, and Zacchia, and under the Nostocales, Anabaena.
So it's mostly freshwater.
Everything's fresh. Well, the Nodularia is brackish, and some of the Lympia is, but everything in here is freshwater. Some of them are brackish marine. So that's the taxonomy. That's not the key. Okay. It's not the key that you have to use to try and identify. So the next section, 25, flip that page to page 29, and you'll see the reference chapter out of the source to treatment book. And that's the dichotomous key that I want to illustrate and demonstrate for you. So, let's leave it at that then, and let me show you what I'm referring to here. Algae. Most of you have looked at the standard methods wastewater. Ever since that was been published, if you flip to the back of there, there's some cartoon illustrations of different kinds of algae, classed as to whether they're drinking water algae or polluted water algae or attached algae. What I'm doing is, okay, this is how you kind of familiarize yourself with algae. That's how I started. And of course, they're cryptic, they're not accurate, but they tell you the various shapes. And so in here, despite what you're seeing in all kinds of environments, normally we see mixed populations, diatoms and green algae and all things, and maybe a few blue-greens. This is what's changed. We've gone from this, which might be a nice diverse population. We're always told in ecology, remember, diversity implies a healthy system. Well, the same thing in the plankton. Diversity implies a healthy system, and now that we have 100% of a microcystis bloom, that's obviously not a healthy system. So, same idea. So this is another one of those. Of course, here's a Aphanizomenon. There's a few blue-greens. There's an Anabaena up there, so on and so forth. But they were behaving themselves at this point. And another one. So when it comes to harmless algae mats and blooms, you can tell a lot. As I've already mentioned, if you pick up an algae mat and it's kind of coarse and fibrous, almost like hair, that's going to be a green algae, a harmless algae. And even though it looks like nice clumps of algae, it looks different than what a cyanobacteria bloom would look like. Likewise, I would often get people sending samples and saying, is this a toxic algae? And it's not even an alga, it's a plant. So things like duckweed can be confused for algae, and it's an honest mistake. How many of you have made that mistake? You don't have to tell me, but people do. So this would obviously be a harmless aquatic plant. But this is not. So when we see situations like this, and what you've all seen, then we start to be suspicious. Especially if we pick this up and it's slippery and slimy and falls out of our hands and so on and so forth. I put this in because Dr. Hans Perl is doing most of the nutrient work now. He's the one working on Thai Lake in China. And in fact, this was taken just last month when he was there. But that's what that lake looks like all year round now. Yeah.
So would you ever see that paint like green or the pea green if it were not cyanobacteria?
It could be yes, the answer is yes.
Yeah. So you can't just go...
No, right, no. For example, the unicellular green, it could be chlorella or chlamydomonas, or it would still look this sort of uniform greeny color. But one thing I have noticed, see how this is not a true, real bright green? Whenever it starts to be kind of a dull green or even a yellow green, that's not a green algae. The green algae, if it's chlorella or chlamydomonas or some of the others, is really going to look grassy green. Even this looks almost dark green. That's another clue, because part of what you're seeing here is kind of those bluish green, you can see almost a bluish green hint to this. But if it's a green algae bloom, it's going to be really bright green. Not an off kind of green, or a real dark green. I know that's hard to, you have to look at a lot of them, but you can tell. Anyway, there's your clues. So when it comes down to it, identification-wise, toxigenic cyanobacteria, and I use the term toxigenic meaning potential toxin producers, because we don't know that until we test for them, are found in 4 of the 7 orders within that Cyanoprokaryonta. The new classification scheme, this classification scheme, defines 7 orders. 4 of those contain toxic or toxigenic genera. I'm going to run through the 3 major ones here. Now, prior to Komarek's work, there was a taxonomic reclassification in 2002 with the Oscillatorias. Some of you have asked me about Oscillatoria and drinking water, reservoirs, planktonic and benthic. Oscillatoria used to be the genus for whether they were planktic or benthic. There's enough difference genetic-wise and otherwise to separate, and so all of the planktic ones were converted from Oscillatoria to Planktothrix. So that's, you probably doesn't bother you because you've been around long enough for that change, but that was the first significant taxonomic change within the potential toxin producers. So any time you get a planktonic or planktic Oscillatoria, that is now Planktothrix. The benthic ones attached are still Oscillatoria. Okay? The other changes that came about with the Komarek work, this one, are follows. The traditional Anabaenas are now using that polyphasic method, are broken down into these 5 genera. The only one that is significant for you is Dolichospermum. All of the planktic Anabaenas that we would have identified as toxic producers are now Dolichospermum. So the Anabaena I'll show you today is Dolichospermum, not Anabaena. I don't like it, but you may like it, so some people say it's easier to say. There's a couple also in this, but not so important. This is the main one. Aphanizomenon has actually been split, and most of the toxic species of Aphanizomenon are now Cuspidothrix. The one up in Klamath Falls is still Aphanizomenon, and it's non-toxic. Aside from that, the only thing you need to worry about, Anabaenopsis is now Anabaenopsis and Cylindrospermopsis. Of course, Cylindrospermopsis is a toxin producer. So those are the main changes. When it really gets down to it, there's not a lot, but they are significant, especially for Anabaenas and Dolichospermum. So, summary. All planktic Anabaenas are now Dolichospermum. This includes the major cyanotoxin producers that produce microcystins, anotoxins, and saxitoxins. In that big book by Komarek, this is the rationale for making that taxonomic change. I just clipped this out of the book on page 792, where he talks about the phylogeny of the genus Anabaena since early 1900s. Contained a wide spectrum of planktic and benthic types, phylogenetic analysis, molecular data, that's the PCR 16S, indicate clear divergence, particularly of benthic mat-forming types in the planktic species. So the planktic species are related to Aphanizomenon and have been separated into the genus Dolichospermum and SWRCB. The original genus Anabaena is basically still up in the air, and you don't want to worry about it. But the Dolichospermum Because down here, see, it says studies of toxicity of various Anabaena species now are Dolichospermum. So he does make that distinct statement. Oh, okay, finally, in summary then, the taxonomic changes for the cyanobacteria, most important, are planktic members of the genus Oscillatoria are now Planktothrix. Planktic members of the genus Anabaena are now Dolichospermum, with a couple in the genus SWRCB and planktic non-colony forming Aphanizomenon, non-colony meaning up in Klamath Lake, that's colonies, are now the genus Cuspidothrix. And so the ones that we have identified as saxitoxin producers are now in this Cuspidothrix genus, except for this Aphanizomenon species grassily is still a toxin producer. So they're both. But Aphanizomenon flos-aquae, the one that's in Klamath Lake, is not a toxin producer. So those are the key changes. So looking at those key orders, just as an example then, the order Chroococcales, family Microcystaceae, contains Microcystis, which is the microcystin producer. And this is a colony of Microcystis aeruginosa, probably the main one. And I also tell people, including you, that you should, whenever you see a Microcystis, there's a high probability that it is a toxin producer, regardless of whether it looks like a typical aeruginosa, which kind of puts you off the hook of trying to tell them whether, it's the different species. There are ones that are more likely to be producing toxins than others, especially aeruginosa, but flos-aquae and viridis and all these others I'll show you can produce. Somebody asked me, can you identify Microcystis that's in an aging, decomposing bloom? Well, here's what a colony of Microcystis looks like when it gets old and decrepit and starts to fall apart. The cells are coming off. At this point, toxin would be out in the environment, but you can still identify it as a Microcystis, but you can't really identify the species. Note what else happens here. Because it's secreting all these materials, these nutrients and proteins, you're getting a lot of bacteria start to come in and feed on the extracellular products. The taxonomic key for the Microcystis, which I won't ask you to worry about today, but it is in the handout. This is the one from China. So we could determine the different species based on this colony and cell size and shape. But here, where are we at? Down here, aeruginosa. That's probably the main toxic ones, but really all these could. I have seen flos-aquae produce microcystins, viridis produce microcystins, Wiesenbergii produce microcystins. So I think you have to be suspicious of any Microcystis. But there are some differences morphologically. If you have nice, healthy colonies, 1 and 2 up here, this is aeruginosa, and down here is what's called Microcystis botrys and Microcystis spermus. And you can see differences in the shape of the colony primarily. But once they start to age and decompose, that sort of morphology doesn't hold anymore. Likewise here, flos-aquae, Ichthyobleba, Novosecki. There are differences, kind of subtle at times. So again, I wouldn't worry about trying to figure out which one they are. I would think these are potentially toxic. And if you're asking the questions, do I test for toxin, then I would say yes. Pseudofilamentosa and Smithii as well. And viridis and Wiesenbergii, same thing. In that order, Nostocales, then, the Aphanizomenon ace, these are the main producers of cyanotoxins. All known cyanotoxins are found in this family within certain producers, like Dolichospermum, which was old Anabaena, So here's a couple of Anabaenas here. This one is probably flos-aquae piece. This is probably, well, this is one of the other species. See, there's a heterocyst here. That's why you know this isn't oscillatoria, because it's got heterocysts. Here's the heterocyst of Anabaena, one there, and one here. The other thing this picture is nice for, notice how mottled these vegetative cells look? Well, that's because they have the gas vacuoles, and that's why they're floating. They're literally gas bags inside the cell that contain gas byproducts. It could be oxygen, it could be nitrogen, it could be whatever waste gas is produced by whatever metabolism. But they fill up and give the buoyancy, and hence you get them floating into the water column. And when light hits those bags, they literally reflect the light. And so under the scope, they look mottled. If you had something that was benthic, it would be uniform, kind of a more uniform greeny color. And that would tell you, well, there's no gas vacuoles as well. So there's Dolichospermum. Here's two species of Dolichospermum, and this is in the samples that are in the back that I brought from Lake Cullowhee in Oregon. 2 or 3 different ones. This is actually a Cylindrospermopsin. 2 species, Crassum and Planktonicum. Crassum and Planktonicum. See the gas vacuoles, you should be able to see, or the, sorry, heterocysts, you should be able to see them. So 2 species of Dolichospermum, and that's in one of these samples in the back that I'll point out in just a sec. And you should be able to see this under the scope. This is just under a normal 200 power phase scope. Now we can count cells. Might not be so easy, but we can count cells in there. Aphanizomenon from Klamath Lake, in this case again. One to point out here, see how big this is? This is one of the akinetes. This is a resting spore. This is not a heterocyst. This is an akinete. And Aphanizomenon has these giant akinetes, huge. Because an individual vegetative cell is like right there. So there's 1, 2, 3, 4, 5, 6, equivalent to about 10 vegetative cells long. And then, of course, if you were backed out, this would be at maybe 100 magnification, you would see the colony. And if you went to 400 magnification, then you see the individual filaments in the colony. I don't know if you've ever been to Klamath Lake, but this is what the harvesters look like on Klamath Lake for AFA harvesting. Each one of these screens for scale is probably about 15 feet, 10 or 15 feet across and about 20 feet high. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9 screens. And they just scoop through the water and lift that algae up onto nylon screens, and these are rollers. The algae rolls up and then is processed or dewatered and then dumped into these shuttle craft, which take them to shore and pumped into cooler tankers, where they're then further processed and made into the algae health food powders that you can buy. Klamath Lake is unique. There aren't very many lakes in the world where that bloom happens every year, and has happened that way for at least almost 100 years now. Ever since people started to settle in the valley, there are reports when settlers used to have to harvest ice off the lake for cooling their foods. The ice would be green at times. Although typically this doesn't come along until midsummer, these blooms. But one of the few places, the only other places in the world that have this consistent same species would be Rift Valley lakes in Africa. And everything's changing, because if you're familiar with, of course, the agricultural practices of livestock, summer livestock feeding in the upper parts of the Klamath Basin, and the agriculture in the lower parts of the basin and clear down into California use significant amounts of water, so the water levels are highly regulated. And now with the federal endangered species and tribal interests, then things like the endangered suckers are you have to control the lake water levels for that. This did and still has a nice population of mine went blank. Big fish. Sturgeon. Some big sturgeon have come out of this lake. And of course it's an important flyway as well. So it is a lake, and that whole basin is receiving a lot of attention for water management now. In the genus Cylindrospermopsis, which is in that same Aphanizomenon AC group we're just referring to, this is one that I think needs more attention here in California and elsewhere. This is the filamentous one, and it has this special tapered heterocyst, or akinete, a tapered akinete on the terminal end, and that's a distinct taxonomy for that genus, and that's the producer of Cylindrospermopsin. We need to be paying more attention to whether we find that in any of your water supplies. The order Nostocales, the only thing I threw this in for is to show you another one of these cyanobacteria that's filamentous, but shows another distinct characteristic of filaments with the sheath. When you enclose these cells in a sheath, and then the filament breaks, and then it keeps on growing, you get this called false branching. So that's another characteristic of taxonomy. But typically, if you see branching in those filamentous cyanobacteria, those are not toxin producers. I don't know of any that are. So just an example of that. And then finally, in the order Oscillatoryales, two families, Microcoliaceae and Oscillatoryacea. The genus Planktothrix in the Microcoliaceae, which is toxic, and the benthic oscillatorias that can also be toxic. And also Lingbia, which is in the same group, because these are, notice, see, none of these have heterocysts, so this is how they're thrown out of the Aphanizomenon AC into the Microcoliaceae, Oscillatoryacea. They don't have heterocysts. So if you see these straight chain filamentous things without heterocysts, then they're probably over in this grouping. If they have the heterocysts, then they're over in the Anabaena groupings. A little bit nicer micrograph to show you how this works. This is the genus Lingbia, both marine and freshwater. See this thick sheath makes them slippery. The filament can actually break, and they still maintain the integrity of the filament. This is an individual cell. How much wider it is than long. That's typical of these Lingbias. And these are big, too. These are the size of a hair on your head in terms of width. And then finally, one that's of some interest here in California, Microcoliaceae genus Arthrospira. This is what used to be called spirulina and is still marketed as spirulina, and this is what's harvested down off of the, Earthrise Farms is the biggest one down in the Imperial Valley. And they're aquaculture. They're not grown in natural ponds. They're taking water out of the Colorado and putting in these big raceways where they grow spirulina and market it earthwise. And this is the powdered material that's marketed. And as near as we can tell, that's not a toxic genus. However, at times they have had issues with contaminations getting in that do produce toxins. So they have a monitoring program for that. Within the Cyanoprokaryonic, the toxigenic members then are in 4 orders, 11 families, 22 genera, about 40 species out of about 1,500.